So welcome everyone to this new seminar. So welcome everybody to this webinar organized as part of the VacCAP project. Today our guest speaker is Patricio, Dr. Patricio Muñoz. Patricio is associate professor and lead the blueberry breeding program and genomics program at University of Florida. And he also director of the PhD degree in plant breeding at University of Florida. Before Patricio worked as an assistant breeder, forestry breeder in a company, and he received his bachelor and master in quantitative genetics and PhD in plant molecular breeding from University of Florida. His research focused on improving culture with a local focus but global impact, and in developing and implementing method, models, and tools to accelerate the breeding process of blueberry. He has released or collaborated on the release of over 10 blueberry cultivar, 4 forage cultivar, and 7 turfgrass cultivars. Currently, is working on developing blueberry cultivar with better flavor, sensorial, and nutraceutical trait to enhance the consumption of blueberry, while still working on traditional crop trait that help producer stay competitive. Today, his presentation will be about volatile, link with flavor, and the status of genetics and method for selecting for this trait in blueberry. Before we start this webinar, I want to remind the attendee to mute yourself in case you are not muted yet, to write questions in the chat, and at the end of the presentation, we will have 10-15 minutes for questions. And with this, I will welcome Patricio, and you can start when you're ready.
Thank you, Massimo. Josie, whenever you're ready. Okay. So let's do this. So welcome everyone. Let me just do a couple more things here so I can see actually my slides. So welcome everyone to the webinar today. As Massimo mentioned, it's part of the VacCAP project that we have. Today I'm going to be presenting a lot of the work that we have been doing over the years, that the University of Florida has been doing over the years, but also the work that we are doing as part of the VacCAP project as well. As an outline of the webinar today, and I think I posted ahead of time this in the social media for people to see what we're going to be talking about. So these are the different topics, but the central point here is flavor and volatile organic compounds, or VOCs, that we call. Okay. First, you know, it takes a village. So just I want to acknowledge my lab. Many different people is working in my lab in these projects in different ways, and and also, very importantly, Charlie Sims that run the flavor panels or sensory panels in here, Denise Tisman, Mark Sende, Annie Ploto, and James Harinuk that actually is running the volatiles for us for the project. Also, importantly, the VacCAP project, you know, and the funding from the USDA that we are able to do the project that we're doing right now, and many people involved here. So why are we here, and why flavor? So first, let's start with this kind of main scheme of the presentation. And first, and it's impossible not to talk about, you know, the healthy components of blueberries, and then one of the things that blueberry have increased significantly over the year, the consumption and production, is because of this strong message and then the research behind of the healthy components of blueberries, right? Secondly, and as a response, or kind of in a circular way, you know, you don't know what is first, you know, the egg of the chicken. The blueberry production has to increase in response to this, or the blueberry increases, and then you have to this marketing campaign behind. Increases tremendously, and is projected to increase even more in the future. With this, of course, is a response also to the consumption. And this is data only from the US, but consumption keeps increasing This is data only back to 2018, but the new data that hasn't been plotted in this way yet is shown also that the consumption per capita keeps increasing for blueberries. Back in the days, and then I know Jessica is attending this. I believe I saw her in the people attending the seminar today. So they started this project of, you know, flavor has to be more important, and then basically somehow they set the basis or the foundation for the work that we have been doing as well. So that was way before I arrived into this lab. Mainly the work that they did, they were able to find out that the flavor was one of the most important components of blueberries, and is the reason that we are here working. Most importantly, whenever this survey was carried, you know, in the recent years, and it was published in 2018 and for both for blueberries and cranberries, you know, the people involved in the production chain of blueberries, you know, growers, nurseries, processors, packers, and scientists as well, all recognize that flavor quality, fruit quality is the most important trait, are the most important traits. And within those, you know, firmness, flavor, and shelf life. Most importantly, Josie keeps popping up, saying that the new people is joining. So I don't know if you can make it for them to join automatically. But anyway, so it keeps popping up in my screen. So most importantly, consumers are able to recognize and are willing to pay more for quality. So this is out of a tasting panel that we run here in the University of Florida, involving around 20 different cultivars last year. And then also this question as part of the VacCAP project was asked, was the willingness to pay. So as the capacity, or as the people enjoys more the fruit that they are eating, you know, they are willing to pay more money for that. So up to this point, we know that blueberries is increasing, consumption is increasing, flavor is important from the point of view of the original surveys, from the surveys to the people involved in the work, and then people recognize and is able to pay more. But what is flavor? So we're going to define flavor in a very simple way. Flavor is taste plus the aroma. And in taste, in this sense, is going to be what the tongue is able to sense, which are going to be the sweet, the sours, the salty, bitter, and then umami. Plus the aromatic compounds that are going to go through the back of your mouth here, and they are going to go through the same sensors that we use, actually orthonasal, to recognize these different aromatic components. These aromatic components is what we call volatile organic compounds, which are naturally occurring components in the case of blueberries. And then we are here to discuss about. Very important, human can distinguish more than 1 trillion different smells. So we have, I'll be talking about, you know, how many volatiles that we have, but basically the many different combinations, and humans, we are able to still recognize them. So again, flavor is going to be defined in a very simple way. Taste, what the tongue can sense, or taste, and then the aromatic components, they are when you chew this fruit, in the case of blueberries, you chew the fruit, and then you break down these aromatic components, they go through the back of your mouth to the same sensors that actually are in the base of your brain. But I have something to tell you here that's very important. Flavor is very complex. It's very, very complex. And a lot of things influence flavor. And actually, for example, let me tell you, culturally, we might, from different cultures, and then we might appreciate flavor of a given food or fruit differently just because of that. The knowledge, if you know, for example, if you have eaten what you are eating in that moment before, you have certain expectations, you know, your brain acts as a model, so it has certain expectation in how should it taste. If it doesn't taste how you are expecting, then it's going to be bad for you, for example. And then experience, and I think this is very, very important. I usually mention about this. Imagine if I cook, I mean, right now, I want you to imagine the best meal or food that your mom or grandma used to prepare for you, right? And in that moment, as soon as you do that, you know, you start salivating and thinking about that dish. Now, even if I prepared the same dish, the best way that it can be prepared, it never is going to be better than the ones from your mom and your grandma. Why? Because this is part of the culture and the experience that you have with that dish. Because it's way more than just how it tastes and what the aromatic components it has, but it's the experience that you have with that dish. So how do we evaluate flavor? In breeding programs, we usually, it's the job of the breeder, but I imagine if as we are, here, it's not just me tasting the fruit. I have a bunch of people here that help me to define if something is tasting good or not. And usually have people from different cultures, so that helps me as well in different ages. The marketers usually are very harsh, I would say, in evaluating the flavors of the fruit, and usually act somehow as a doorkeeper of what fruit moves into the retailers. And I have something to say about that later. And then we have sensory panels. And with sensory panels, I will say that they're very accurate to define, well, it depends on what sensory panel you're running, but let's say it's to define relatively if one given individual or genotype is better than another. Sensory panels are great because it provides a lot of good data to understand what is happening with the fruit, how people is understanding and tasting and is experiencing the fruit. However, they are not very high throughput, they are costly, and they are time consuming. Okay, so I just told you that flavor is composed of these two things. So we have the taste, and then we have the volatile organic compounds or the aroma, right? This is provided by the volatile organic compounds. But what are volatile organic compounds? And what do we know about them? First, what we know is that the volatile organic compounds are present in Vaccinium species. And this is out of some work that we did in 2020, that we published in 2020. And this is present in many different, in almost probably all Vaccinium species. At that moment, it was very easy to compile all the research that's been done. And I think there is a lot more now, and it will be a lot more difficult to compile every single paper that is published. These are naturally occurring metabolites, that occur in the fruit, naturally occurring in the fruit. And then basically they come from different metabolic pathways. We also know that the volatile organic compounds in Vaccinium species, they have been studied since 1967, sorry. And they have been studied using different methodologies and on different species of Vacciniums, different genes. We know very well, and not just from blueberries and from Vacciniums, but also from many other different species, that these different organic volatile compounds that we see here on the outer ring, they elicit different sensations when you try the fruit. So some of them, for example, are going to be more fruity, citrusy, floral, green, or minty. And then these are the very bad ones, I would say, in the case of blueberries. And then I'm pretty sure that these are the ones related to some of the, what we call the wet dog or wet sock category that we have in blueberries. That's pretty disgusting. Those are the ones that you put in your mouth, you try, and then you usually end up spitting them very, very quickly. And my blueberry colleagues, I'm pretty sure that right now they are laughing because they have experienced this. These volatile organic compounds can be classified in functional groups as well, like alcohols, aldehydes, terpenes, aromatic esters, and ketones. The issue with this, and some people doesn't like this because, for example, some terpenes could be classified as alcohols as well. So it's not a pretty good classification, I would say. And some people don't like it, like I said. So a better classification could be, for example, if we use this classification of the origin, where do they come from? For example, we have, 10 to 11 volatiles that come from the branch chain volatile pathway. We have 28 to 29 derived from the fatty acid pathway. We have 11 from the terpene pathway and 7 from the phenylpropanoid. And we have around 3 that they come from primary metabolism, and also they are unknown. This is a reduced list, around 60, that are the ones that we usually classify as the ones for sure we know that exist in blueberries. But with the new methodologies that we have been using, we are finding a lot more than this, and I'll be discussing that in a second. We also know that volatile organic compounds are important for sensory, for what people taste. This is a study that we just published this year, that it shows that actually out of the overall liking of the fruit, 56% is explained by the volatile organic compounds, a difference of 39% that explains by sugar and acids. So somehow, you know, since the beginning of reading of blueberries, we have been focusing on sugar and acids. And then volatile organic compounds, well, have been playing a role, indirect role, because you like them, they might have higher organic volatile compounds. You know, you're not selecting directly for that. But nowadays that we know that they are a lot more important, you know, we do this in a direct way. So how they are analyzed, and I know I'm talking a lot of these things because it's kind of general education about this. So for sample preparation, blueberries or a fruit can be used as a whole. I don't think you're gonna get much about that. They can be chopped, they can be smashed, and also you can use juice. For the extraction of these volatile organic compounds, you can use dynamic headspace, distillation, static headspace, and solvent or liquid. And then for quantification, the basic way to quantify these volatile organic compounds in different ways is GCMS, or gas chromatography mass spectrometry. And I know nowadays, for example, there's different ways, there is different levels of this GCMS, but in reality, that's the way to quantify them. Regarding the different methods that I was just mentioning before, you know, we have over the years, and then the study that we did in 2020, we found out that, you know, there is not a huge preference for one or other methodologies over the year. But recently, we have seen that more dynamic headspace and static headspace is being used. Okay, so now we know what volatile organic compounds are. You know, where do they come from? Now, as breeders or geneticists, it's very important for us is what is the genetic architecture? And when we talk genetic architecture, we mean what is the genetic control of these characteristics, you know, or the heritability? What are the number of genes controlling these characteristics? The location of this gene, the gene action, additive, dominant, epistasis, and also the effect size, very important. So first, let me tell you, this is a graph of the heritability of a subset of these compounds, of these volatiles. And then from our point of view, this is considered medium to high levels of heritability, which is pretty good if we wanna do improvement for them. Very, very important here, and I need to do a pause to mention that we are only able to do this work because nowadays we do have a genome for blueberries. And actually nowadays we have a few genomes for vaccinum species. And as part of this project, very soon, hopefully we're gonna be releasing a lot more genomes and also the pan genome. But this is without the genome, the information using the exactly same data, and then using the information with the genome. Of course, now we know the different chromosome, the homologous chromosomes is able actually for us to find where is the location of the different hits of the characteristic whenever we carry this GWAS analysis. More importantly, in the case of volatile organic compounds, when before it changed completely the way that we believe that these traits were controlled genetically. You know, before the genome, we kind of were thinking that this could be, you know, a quantitative trait controlled by many genes, but in reality, with the new genome, and again, the same data, we're able to find out that all these, you know, pieces of genes or genomes that they were scattered all across in different random way, in reality, they belong together. And it gives us a much better picture that these traits are controlled by a much simpler way. So in case we can call it monogenic or maybe oligogenic in this sense. So very importantly, as part of the new VacCAP project, this is as part of the study that we did in 2020. And then we were able to find out, for example, what are the gene regions important for eucalyptol, a very important volatile for blueberries, because we know that this is a deterrent of liking. With the new data, actually, we're able to validate, and now we're going to be putting this together, and hopefully we're going to be narrowing down even more where these different characteristics fall. Now, this is part of the VacCAP project, but we are using genomic data that we do have in our program here. But very soon, as part of the VacCAP project, we're also going to be releasing the set of markers that is going to be used for the future of analysis in blueberries and cranberries. Also, with this new data, now we are able to find out the location, the genomic location of other characteristics that are very important. For example, methyl isovalerate. That is another volatile compound that we know is very important to give, these fruity characteristics in not only blueberries, but in other fruits as well. And at the same time, we know that when we want to study these more complex characteristics or more complex modelings, for example, if these traits are dominant or epistasis, and in the case of blueberries, that is even more complex because it's a tetraploid species. Fortunately, through the years, we're being able to work actually to define the models for this species. And now, by putting all this data together, the old data with the new data, we're able to identify better, what are the areas of the genome, not just the additive portion, but also where are the other parts of the genome that are interacting in different ways to provide the control of these volatile organic compounds. So as geneticists, now we know a lot more about this box, right? We know more or less where they are. We know, if they are, how much control do we have from a genetic point of view. And we know how many genes, more or less, or how many areas in the genomes are important for the control of these volatiles. But as breeders, we want to know if we can predict them. That's very important. So first, as part of a study that we just published this year, I just want to show you, in this study, we use all the data that we have for sensory panels and also metabolites back to 2020, I believe. And one thing that I want to highlight here is like different metabolites or different volatiles that, they are not very intrinsically connected. And then for biochemistry people, this might be, obvious, but what we were worried that actually the volatiles could have a lot more influence and it could be a lot more overlapping on deciding, if in order to detect them, you need to be sweet or you need to be acid or so on and so forth. But actually we see the sweets and acid, they are not very, very well connected with all the networks of volatiles. So this is good for breeders because that means that we can increase liking of the fruit independent of or somehow independent of the levels of sugar and acids. In this study, we also were able to publish, data from blueberries and also from tomatoes. Unfortunately, we didn't have data for blueberries in genomic data for blueberries. So we used the data of tomatoes actually to quantify or to estimate how much we can predict using this genomic data, but also the metabolomic data. Now, in this study, we show that this metabolomic could be a lot better to predict, for example, over liking. Now, this is somehow expected. Why? Because, you have the gene, then you have the expression, the transcript, and then you have the realization, the metabolic, that all this transcript make happen, the proteins and metabolites, and proteins, enzymes, and then, all this, gets driving the metabolites. So the metabolites are a lot closer to the final phenotype, of course. And the final phenotype is going to be a lot closer to what the people is sensing. So we're expecting that the metabolomics is going to be better in the prediction. Now, this data set was very small, I will say, for our genomic study. So, but it's whatever we had available at that point. I expect that by increasing the amount of data, we are going to be able to do a lot better with genomic. But remember, liking or sensory traits are very, very complex because of what I was discussing before. In this sense, we also try different methodologies. And one of the methodologies, one of these AI or machine learning methodologies, like gradient boost machine, is one of the perform the best. Now, like I mentioned before, for us, it's very important to be able to predict not just the sensory, but also the volatiles, because the idea of just doing metabolomics or measuring every single metabolite is also expensive. But can we use genomic data also to predict these metabolites? We should be able to do it, right? Because I just showed you they have a high genetic control. So in this case, we have two populations, so the population 1 and 2, and we use different methodologies to validate this. So we have, for example, cross-validation here within the population 1. So we have another scenario that we have direct genomic selection. We have another scenario where we did a GWAS. Then the GWAS were combined with genomic selection, for example, using these markers as a fixed effects in these models. And then we used to predict the population 2. Or another method, the last methodology in red that actually is used traditional marker assisted selection. So we have a discovery phase, and that was used in the population 2. And we were able to find that in most cases, actually, the direct marker assisted selection, it was performing very well. Like, for example, you see here, here, here with 2 non-unknown. So in this case, it's telling you that just not only we're able to estimate that the genetic control for these characteristics falls in a very, very few spots in the genome, but also when we use these markers in those spots, we're able to predict them very well in an independent population. Independent because these populations of Southern high bush, they're all related somehow. Now, in other cases, and this you're gonna be probably already thinking, it's like, wait, that's not the case. I see other cases where it's not the case. And for example, in the case of 2-hexanol, where we see that the marker assisted selection performs worse than genomic selection, mostly indicating that these characteristics are gonna be a little bit more complex than, for example, the 2 non-unknown here. Now, probably you're asking at this point, okay, what are the volatiles that matter? And the answer for that question is going to be, what is the sensory trait that you are talking about? Are you talking about overall liking, sour intensity, sweetness intensity, or flavor intensity? Depending on different ones, and we have a lot more, right? But depending on which ones you are looking for, you're going to be looking at what are the different volatile organic compounds that are more important. Of course, we're always going to have, sugar and acid. That's very important characteristics. So here we have the level of importances in the x-axis, and we have, kind of the positive or negative correlation with liking in the y-axis. So at this point, I hope that I have convinced you that volatiles are important, that we know a lot more than we used to do. Now, not every volatile organic compound that exists in the fruit is going to give the aromatic sensation to the people. This is very important to understand for the next part. So not every volatile that already exists in the blueberries is going to give you a sensation of aroma, right? So there is going to be different groups of volatiles that some of them provide this aromatic sensation, some of them that cannot. So a study that we started some years ago, we were asking the question, okay, we are able to find out that these blueberries are different. They have, these aromatic components. So can people detect that? When I say people, can consumers detect that? And now the second question is like, do they like more this aromatic than the non-aromatic? And the third question then is like, so then where this aroma come from, right? And then obviously as breeders, like we want to predict everything. We say, can we predict this, right? So let's see the results for this. So this is also as part of a study. This is already in bioarchives, but it has still under review. So here we have the aromatics in blue, and then non-aromatics in reddish or brownish, whatever. And then let's just focus in the overall liking. We were able to find out that the people not only is able to detect what breeders usually classify as aromatic, but actually, but actually they like them more. So this is a statistical significance here. And then whenever we plot all this data, we're able to see that what we breeders classify as aromatic, this aromatic dot here, a point comes from the breeders, this point comes from the public, from the consumers. So this is out of tasting panels that we have. And at the same time, we're able to find out already, give us a first hint of what are the volatile organic compounds that might be important for this aromaticity found in blueberries. As an overall, when we look at the characteristic of these aromatics here in the lower ring, compared with the non-aromatic blueberry individuals, one of my students just asked me, we need to find a way to change, to indicate that when we say aromatic, it doesn't mean aromatic in a chemistry way. So then that's why I want to be very explicit about this. These are aromatic, a set of aromatic genotypes of individual. This is a subset of individuals that do not have you cannot sense the aromatic components, right? So when we look at them as an overall, we see that the major difference is in the terpenes. So the aromatic individuals or genotypes, they have higher concentration of terpenes. Now, this is very important. Remember, I told you that the aromatic ones are liked more, but in reality, when we look at, the levels of sugars to acid, there is no much difference between aromatics and non-aromatics. But whenever we look at for example, the sum of the terpenes that we believe that they're the ones that are providing this aromatic sensation to the blueberries, we see also a positive correlation with the sum of the terpenes. And then we see now that it's very easily how do they group aromatics and non-aromatics. As I mentioned before, for us, it's very important to predict these characteristics. So we're able to, using classifications, curves with threshold models as well, we're able to use some of these terpenes to be able to classify if an individual is going to be aromatic or non-aromatic. So a lot's been done. A lot of progress, I will say, toward the understanding and the use of this in breeding. And there is a lot to do still. So I will say that as part of the VacCAP project, we're going to be able to build a lot more resources, not genomically, like I mentioned before, with the pan-genome that is coming soon, with the chip array or the array that we're going to be using, the molecular markers that we're going to be using to carry the research. But at the same time, we're going to be in one point, pouring all this information about volatile organic compounds to the public whenever we're able to finalize the analysis. Very importantly, in the case of volatile organic compounds and flavors and sensing, is that we really need to understand, for example, linalool is a volatile that's correlated with positive liking, right, with liking. So is more best? Is always more the better? We don't know that very well. So for that, we are using actually threshold models to understand if that's the case and what is the point of basically the diminishing returns. So we stop pushing for that given characteristic, in this case, the volatile. Validation in different ways, in silico, in situ, in planta, and through using gene editing as well. So all the different things that we are working. And very important, high throughput methodologies. High throughput methodologies not only for actually being able to quantify these organic volatile organic compounds, but at the same time to be able to have a high throughput way to understand if the fruit is liked or not. And that's where the artificial intelligence prediction is coming from. And I'm going to be mentioning more about in a second. But I will say very importantly, I think right now we have built enough data to reliably tell that there is no a single blueberry that we need to be breeding for with a given characteristic. Every blueberry, with the exception of the very, very acid ones, it has some people that likes them. And if you are breeding for, the average people, the average consumer, increasing sugars and acid might be the response. But there is a lot of market there, a lot of market, and then a lot of possibilities. And it's incredible the amount of variability that exists in blueberries and which directions we can go. Basically, it's just defined of how do we connect with those retailers, with those chefs, and with the people that sit in the blueberries. Now, regarding this artificial intelligence, I was just want to give you a snapshot of what we are doing. So remember, I told you that the sensory, collecting the sensory, and even if I ask you, people that understand very well about, the liking of the fruit or something that is good and something that is bad, whenever we look at the extremes, it's very easy. Something that is bad, something is good. But in the middle, it's very difficult to say if you like it a 5 or a 6 in a scale of 1 to 9, or if you like it a 40 or a 70 in a scale of 1 to 100. So in this sense, we are starting and we have some very preliminary data, and we hope that this could help us as well, basically to evaluate how much you are liking the fruit by just doing this emotion collection with artificial intelligence and deep face. We know that plant breeding works, and this is a plot I always show. So this is the firmness of the fruit. This is the year of release of different cultivars and how, plant breeding and management as well has increased these are different cultivars, by the way. In red, northern high bush, in blue, southern high bush. And how plant breeding, with management has increased the firmness of the fruit over time. We hope to do the same things with volatiles and the liking of the fruit, because that's going to be beneficial, not only for breeders, but for also every person in the chain of study for blueberries. How we're going to be doing that? This is how we run our breeding program here. Right now, we are using, metabolites in these parts here, and also sensory panels in this part here. But we expect that we are able to do high throughput methodologies like I'm hoping that we're going to be working in the future on. We can move those for earlier stages in the program, and so take full advantage of all the diversity and variability that exists in the program. I know this is being recorded, so I think it was very important for you to know. So most of the research that I talked to you today, it comes out of the work that we have done in the lab in different years and with different people, and also the research that was done in the lab before I arrived, mainly by Jessica Gilbert, that is here today. So with that, I'll be happy to answer questions and listen to your comments. And I just want you to understand that I'm here providing my vision of the way we see the work with flavor and volatile organic compounds, of the vision of a breeder, basically, and not a specialized person in any of these different methodologies that I talked to you about today. So thank you.
Great, thank you, Patricio. Great talk. So if anybody has questions, you can post on the chat, or you can unmute yourself. I will just start with the first one from Anne Plot on the chat. So by selecting for more volatile, is there a possibility that it will be compromised anthocyanin and the flavonoids, Patricio?
Yeah, so, and that's a good question. The short answer is that I don't know. I have no idea. But we have a large project of in anthocyanins as well. And I can answer you that question probably by the end of the year. The second question that you say, how much flavonoids will affect taste and flavor? The preliminary data that we have, it says that it will not. We do have some, specifically anthocyanins, I will say, high levels of anthocyanins for data, the preliminary data that we do have, it does not affect the flavor. Now, maybe that's up to the levels that we're able to find in the individuals that we're able to test in sensory panels, right? So it doesn't mean that this is an absolute answer. So we just need to keep working to find out if there is going to be a point that is going to affect.
Yeah, another question, what type of population are you using in the study?
Yes. So ERA, what we use in our program, so this is a breeding program, so which is a great advantage. We have the research inside the breeding program, right? So like I show in that in one of those slides, now let me try to go back there. Whenever we plant the stage ones or the nurseries in our program, we have access to 20,000 different segregants, basically, right? And then at the end of that process, you know, we do this every single year. Every single year, we also select 2,000. Every single year, we also select 200. Every single year, we select 20. Every single year, we release a cultivar. Or some years we do not, in reality. But most years, I would say we release 1 or 2 cultivars. So we use the breeding population to do this. Why? Because we want whatever we do to be applied to breeding, right? So we do not develop with very specific exceptions. We don't develop specific populations just to do the work that we do. Now, saying that, and also it's very important that Southern High Bush blueberry, it was originated here at the University of Florida. And most population that exists across the globe, they are related, you know, 1, 2, or 3 generations removed basically from the direct pedigree of our work here. So then we expect that our work could be applied to other Southern High Bush programs as well.
So another question from Jessica. Great presentation, Patricio. Do you have any thought on how model the way how to model the way individual volatile act together in the matrix to results in additive or canceling out aroma perception?
That was in the future, Jessica. Did you see that? We do have some ideas, but at this point, they are just preliminary. You know, I think as part of my PhD, I work a lot in non-additive models. So I do have some ideas on how we can model these things, but we needed more data. And then out of what we have done in the breeding program, plus what we are doing with VacCAP, I will say that finally we will have enough data to answer a lot of these questions. So we are starting to do a lot of this analysis now. But basically the traditional way, you know, multiplicative way, you know, Hadamard products, for example, matrix and doing epistasis, you know, those are the things probably traditionally that we're going to be testing for sure. Maybe some machine learning, artificial intelligence, or non-additive and non-linear modeling probably also is going to help in that way as well.
So a question from Luis, great talk. Could some sort of selection index for Vox be constructed? How could this be incorporated with other traditional trait, yield, disease-resistant firmness?
So Luis, this is a good, very good question, you know, and I think obviously you're thinking of as a breeder. We have tried and iterated a few different ways. You know, for example, and again, in order to do all this, you need the data, right? So the data is the basis, and then I'm very data driven. So then we are building a very large data set for these different things. For example, you could combine, if we saw the terpenes, you know, we know that they provide aromatic. You can get more aromatic out of the fruit with the terpenes. But in reality, maybe your breeding program is not going to be only about that, right? We don't do just that. So maybe, well, that could be one way you will be restricting yourself to find great genotypes that have great characteristics, but they are just not aromatic, right? Now, on the other hand, maybe you have, if you have enough data for liking, for sensory panels with liking scores, you could potentially, you know, try to predict liking. You know, like the same way that we do predict breeding values, you can predict liking as the characteristics. And what do you use there? Well, you have to use everything that you have. And that's the way we do it. So you use the genomic, use the pedigree, use the phenotypic data, you use the metabolites. And you combine all this together actually to get one score that is going to be the one that you're going to be using. Now, how do you combine out with all the other characteristics that we use and you select for? You either use it as a threshold for selection, or you mix it up in the selection index. So that's up to you on how you're going to be applying this information that we generate. That's the part I really like to talk about.
Okay,
it's
so
not very fun for me that I'm talking to myself here.
So any other questions?
I have a quick question.
Go ahead, Josh.
You mentioned that consumers are willing to pay more. I remember that slide in the beginning.
Yep.
How is there even a market for that though? Saying this blueberry is sweeter or more aromatic than the other. Right now, blueberries are just sold as blueberries, right? So is there a mode or a pathway to doing that at the moment or in the future?
Thank you. Can you please identify yourself? I don't know who is asking the question.
So this is Josh Anderson.
Hey, Josh. So I will say there is a market. There is a market. The market might not be explicit, right? But there is a market. Now, some things that we have experienced in the blueberry world, I would say, and you hear these stories once in a while that, you know, some genotypes are not accepted in the retailer anymore, you know, and then basically they sell as a second, you know, class blueberries, and then, or they just they go to processing because they don't accept them as a fresh fruit. So I keep telling my producers here is like, it's not a matter anymore of having better quality. The question is, if you do not have the quality, you get excluded from the equation. And that's the problem that we're facing nowadays. Now, that's one thing. The second thing is consumers are willing to pay more for quality. That happens every day with all our lives, everything that we do. Why do you go to a more expensive restaurant when they serve, you know, a very tiny dish? Because you're willing to pay for quality, right? Now, the same thing happens in the fruit. It's just a think about the marketing. And, you know, Jessica is just telling us, Spencer is just telling us the example of the sweetest batch, right? That's one example. People is buying that, you know, is making a difference. People is willing to pay more for quality. Now, how do we define quality? In the blueberry world, you know, before the sweetest batch, it was defined as like jumbo size of fruit, but it's not the solution. You know, not every fruit can be jumbo, and not every consumer likes jumbo fruit. Actually, I just read this blogger that posted the other day, you know, complaining that the jumbo fruit are kind of a mutant of fruits or whatever, you know, and then she really disliked them. And she went over and over how this fruit shouldn't exist. Anyway, people is paying. This data that I got, that we got, is out of the VacCAP, and it's out of tasting panels, you know, out of 300 people that taste 20 different varieties here at the University of Florida. And the same data is going to come from the studies that we're doing in Oregon for over 2 years. So we're going to have a lot more data to show that actually people is willing to pay more to have a better experience with the fruit and the food that they are eating.
Do you ever see the future of marketing blueberries by cultivar?
Hopefully not. Yep. I don't want us, I mean, that's my personal opinion. I don't want us to be apples, because whenever that happens, there is going to be a very limited space in the shelf. And then there is going to be a fierce competition for whom gets in the shelf. And then in reality, the issue is going to be that people get stuck with some varieties, very old varieties, and then doesn't allow for some innovation in some cases. Now, Jessica Gilbert is pointing me out here that blueberries are already sold by cultivar in European countries. Jessica would say yes and no, because in reality, usually the name is in the back of the clamshell. In some cases, when you buy it, you have to flip it if you know what you're talking about. They are not marketed by variety, like apples are, I would say. But you buy in the clamshell, some cases, very small, they say what cultivar they are. In other cases, the name is in the back of the clamshell. In other cases, it's in the top. So it's not very consistent. And still, I don't think they are marketed as those apples. You're still seeing the label in the supermarket, it still says only blueberries. When, for example, in apples, it says Fuji apples. You know what I mean? So I hope that blueberries in the future get marketed by quality. It's a different thing, that we are able to provide a quality score for the blueberries that you're going to be able to eat. And actually, also, there is examples of that in some European countries, and specifically in the UK, where you can find the blueberries in these different categories. If you want blueberries for fresh consumption, or if you want blueberries for smoothies, you can say maybe 1 pound, British pound, in one or the other, for example.
Yeah, thank you very much. I want to just add one thing to what Patricio just said, since has been part of the discussion when we were working on the grant. So I guess one of what Patricio said at the beginning, one of the issues in this phase of blueberry production market is consumers don't buy blueberry anymore because it's a novel product on the market. They are on the phase where they want to buy blueberry because of the quality. The phase where probably the market is today in production is that we need to push for having more stable quality. What Patricio was saying, if you don't have the quality, you are out of the equation. So at this stage, it's not much, a specific flavor, a specific aroma yet, but at least getting the quality. And then probably the next phase could be also specific aromas, but at least at this point, the quality. And then the way that I learned when we talk about willingness to pay, we need to think about willingness to pay not only as the willing to pay more, but also as the willing to buy again. So there is a paper again by Jessica in the past that was pointing that only 20–30% of the consumer buyer are consistent buyers. If you can develop a strategy or come up with a way for those blueberry buyers to be more consistent buyers because you have more uniform quality, so somehow you increase consumption independently of increasing they pay more. You should interpret the willingness to pay also as a willingness to buy again.
Now, I think this is an excellent point, Massimo. And then unfortunately, we all have faced, when we buy blueberries and then they're mushy, soft, and they taste, they are super acid. And then obviously you will not go back and pay again to buy those blueberries. There is a talk that Felicidad from the UK gave some time ago that, indicating that imported blueberries into your country are beneficial to maintain that shelf, the fruit in the shelf, some people repeating and buying, but it's important that this fruit also have quality. So while we focus a lot in the local, improving our blueberries for our local producers here, it's important that there is also improvement for fruit that comes from overseas, that is 20–30 days or sometimes more depending on the country, because otherwise that's going to be damaging, the repetitive buying of fruit.
Any other questions? As I said, either on the chat or you can unmute yourself. While I'm waiting, I want to just thanks Patricio for the great talk and conversation, everybody for attending, and the extension team for organizing the webinar. And just a reminder that Michael Coe just sent an email to the participant with a survey that we can capture your inputs.
Thanks, everyone. It's great to see many names. I don't see faces, I see many names that I recognize. So I don't see any further questions. So
Yep. Thank you, everybody. And great. We'll see you next webinar. Thank you.
see you soon, some of you.